Oh, it's a decent size.
No, I'm sorry. It's like 20. It's not a huge. I mean, it's right. It's not a huge needle. It's just a lot of liquid. Like, this is.
It's a half a mil. You're gonna have a mil.
Which for regular more sexy. Like that. Gotcha.
No.
Really? No, maybe it's because I don't look in that direction. Oh, hey!
You were like, I'm gonna watch.
Now,
Ooh, it's a big
these are
packet.
in my door. That's 2 weeks ago. Because then I went on quarantine. So.
Oh, I'm glad they're not double sided. I was like, that's gonna be
No,
really
no, no,
thick.
no, no, no,
Fixing
no,
both
no,
sides.
no.
That makes it so much better. When's it due?
I think you guys should be able to get that stuff done within 2 weeks,
That seems reasonable.
reasonable. So today's the 9th, so the 23rd. Tell you what, 23rd tentatively. Oh, I forgot to talk to the farm today because
I forgot to-
they wanted to, farm folks want to reschedule the exam. And I forgot to mention that today. I may have to do that by email. Okay. No worries.
Question for
Yeah.
the summary paper. How
You-
long can we extend it?
Yeah,
Like,
you
when
asked
do you want
about
it by?
extending that. I would like it before the start of finals week.
All right. Say I'm like halfway done with it. Not that I have. I'm playing catch up.
Your tests are great. You need to figure out the curve. Oh, no. These, Amanda, are
Thank you. I didn't think
handouts.
so.
Okay. We were talking about the degradation of amino acids. We're now on to the subject of the biosynthesis of amino acids. And I don't think we talked about lysine degradation, did we? I think that's one amino acid we left out, if I'm remembering correctly. We didn't talk about the degradation of lysine. Yes. So before pretty sure we didn't get to lysine, which we can do easily because we talked about alpha ketoadipate, because lysine is degraded to alpha ketoadipate. So if we just talk about the degradation of lysine to alpha ketoadipate, then we'll be home free. So let's talk about that, then the last one, and then we'll be good to go. Okay, so let's. Homework, actually homework. Okay, so, what is here? Okay, so lysine looks like this. So that's lysine. So we have alpha, beta, gamma, epsilon. So the epsilon amino group here. And the degradation of lysine is going to take a very strange turn because you're going to combine this with alpha ketoglutarate. Of all things. So that's alpha ketoglutarate. You're going to put these two together. And then you're going to give this reductant. So this is interesting. You have NADPH being used for a degradation pathway. And you're going to make this molecule that's a combination of these two. So there's your alpha ketoglutarate. And here's your lysine. And this molecule is called saccharopine. I don't know. I don't think so. Didn't make it into the handout. SACCHAROPINE, saccharopine. And essentially, if you take lysine and just attach the epsilon amino group to the keto group here, keto carbon right here, there's alpha ketoglutarate, you get saccharopine. I am not altogether sure. Really not. But, and you know what? There's also water. This? Yeah, H plus. Well, what you do with saccharopine is going to, so take a look at this with me. What do you have here? Who's that? It's alpha ketoglutarate with an amino group. That makes it glutamate. So you're essentially going to get rid of this, and you're going to have this piece that's left. So the next step essentially is going to take saccharopine. And I'm not going to draw all that out. And you have an enzyme called saccharopine dehydrogenase. And saccharopine dehydrogenase is going to take water, and it's going to take an electron acceptor. And you're going to get reduced plus glutamate. And from this, you're going to get this comes from the water over here. And this is called alpha amino adipate semialdehyde. Alpha amino adipate semi-aldehyde. And this is saccharapine dehydrogenase, this enzyme. That's the name of the enzyme. And then all you really need to do is to oxidize this, and you will make the carboxylic acid. So you have the alpha-amino-dipate semialdehyde dehydrogenase, which is going to oxidize this. This enzyme comes in a couple of different isozymes, so it can use NAD or NADP. And now you convert this to a carboxylic acid. Because you've been oxidizing aldehyde, you get a carboxylic acid, right? And that, ladies and gentlemen, is alpha-amino-dipate. And we talked about how to break down alpha-amino-dipate already, but essentially those 3 steps where you make saccharapine, then you oxidize saccharapine, you give up a glutamate, you get the alpha-amino-adipate semialdehyde, you just convert it into through oxidation, you convert it into the acid form, you make alpha-amino-adipate, and then alpha-amino-dipate is converted into glutaryl coenzyme A and so on and so forth. And that goes through the rate oxidation pathway. So that is how you degrade lysine. Those 3 steps, you make saccharapine, you then take saccharapine, you liberate the glutamate using saccharapine dehydrogenase. You make the aldehyde, you then convert the aldehyde to the carboxylic acid. All right, that's lysine degradation. Ain't it grand? Well, we talked about breaking those things down. Let's talk now about building them up. And again, you're just responsible for what happens in the human body. All those glorious pathways in bacteria that I had to learn, you don't have to learn.
We're talking about bacteria.
Yeah,
Right.
but you just need to be able to group the amino acids in the families and say what goes into them. So we're going to say there's a glutamate family, there's a aspartate family, and so on and so forth. Give subtitles a try. I don't think so.
Okay,
Oh.
I don't even want to see this. Right. Some of the words you said.
Oh, no, it's doing it.
No, don't turn it on. Very good. Okay. So in our bodies, we have essential amino acids that we need in our diet. We have other amino acids that you need when you're a kid because you can make them, but you don't make enough of them when you're growing. And then we have those that are just drop dead, non-essential. Now, if we look at the non-essentials, alanine comes from the transamination of pyruvate. Aspartate comes from the transamination of oxaloacetate. Glutamate comes from the transamination of alpha-ketoglutarate. Serine, you can make from glycine. Well, actually, you can make serine from pyruvate. You make serine from cysteine. You make serine from lots of different amino acids. Asparagine, you just make from aspartate. Glutamine, you just make from glutamate. So these types of reactions, no big deal. So these here, proline is made from glutamate. Lysine is made via the lysine cleavage enzyme pathway, or you can make lysine from serine. Cysteine, you make from methionine. Arginine, of course, you make from the urea cycle. The tyrosine you make from phenylalanine. So these are conditionally essential. They're required to some degree in people that are growing. These over here, though, we can't make. We need these in our diet, and we need them, but bacteria make them on their own. So you need to know how these are made right here. These, you don't need to know. But you just need to know the barrel outlines, and the barrel outlines go something like this. So histidine is off the beaten path. So histidine, as you can see, is essential. Now histidine as a pathway is interesting because it interfaces with the synthesis of nucleotides. One of the intermediates of the histidine phosphatic pathway is an intermediate in the synthesis of nucleotides, purines in particular. So histidine, the way it's made is just weird. It's made from ribose 5-phosphate. Very weird. The aromatic amino acids are made from erythrose 4-phosphate and phosphoenolpyruvate. They're made from erythrose 4-phosphate and phosphoenolpyruvate. The erythrose 4-phosphate gets made from phosphoglycerate. Phosphoglycerate is a glycolytic intermediate. Therefore, these two are made from glycolytic intermediates. Lysine, cysteine are made from serine and bacteria. And in humans, lysine is made from serine. We talked about that already. That's what we do. Alanine, valine, leucine, isoleucine are made from pyruvate. Not just pyruvate, but pyruvate is a starting material. You need to know alanine, you don't need to know these. This is the aromatic group. Tryptophan is in a pathway all by itself, but there is an intermediate that these will share. And that is the arrow group of genes that encode the enzymes that make aromatic amino acids. Glutamate and aspartate are made from these Krebs cycle intermediates. The aspartate family includes K-mint, K-mint. Lysine and isoleucines in there too. Methionine, asparagine, which is N, and threonine. The threonine interfaces with the branched chain amino acids. The glutamate family consists of glutamine, choline, and arginine. Glutamine, choline, and arginine. You know how glutamine is made. You know how choline is made. Don't need to know how arginine is made, except for how it's made in the urea cycle. Bacteria use a different pathway to make arginine. That pathway you don't need to know, but you need to know it's a member of the glutamate family. The aspartate family, glutamate family, pyruvate family, aromatic family, histidines on the left on its own, and the serine is made also from 3-phosphoglycerate, which then gives rise to lysine and cysteine. So this table very nicely shows you these amino acids. And if there's a star next to that amino acid, you don't need to know how it's made. If there's no star by that amino acid, you do need to know how it's made. So you need to know all these except for arginine. And it's not essential because we make it via the urea cycle. Cysteine is made from the breakdown of methionine. That pathway you need to know. So these two are interchangeable. These two, but then these don't need to know. These don't need to know. That we need to know. None of these, none of these. Tyrosine is made from phenylalanine. That reaction you need to know, but how these are made, don't need to know. Okay? Are we clear on what you need to know? Okay,
The stars, we did not.
stars you do not need to know, except for tyrosine, you'll need to know.
Arginine, we need to know the family, but not need to know how it's.
Right, you need to know the recycled way it's made, not the way it's made in bacteria. Okay, first, let's take a look at 3-phosphoglycerate and how serine, glycine, cysteine are made. This pathway is pretty straightforward. So 3-phosphoglycerate, first thing that you do to it is you get rid of this here because you want to put an amino group there. How do you make an amino group? You make a ketone out of that, so you oxidize. The phosphoglycerate dehydrogenase oxidizes it. You have a ketone there, now you have 3-phosphohydroxypyruvate. You just add the amino group here. So you have phosphoserine aminotransferase. Can you guess what cofactor that enzyme has? Aminotransferase, pyridoxal phosphate. All aminotransferases, transaminases, have pyridoxal phosphate as their cofactor.
Can you write pyridoxal phosphate on the board so I don't spell it? That would be super wonderful.
Come on, slowpoke. Okay,
Yeah, I would not.
pyridoxal phosphate, and it's derived from dietary vitamin B6. And deficiencies of vitamin B6 cause the 3 Ds. Diarrhea, dementia, and dermatitis. The 3 Ds, that's what deficiencies in vitamin B6 will cause. Pyridoxal phosphate, which is abbreviated PLP. B6 is the form that's then converted into pyridoxal phosphate. All right, so that transamination using glutamate as the provider of the amino group, glutamate is converted to alpha ketoglutarate. And now you put that amino group into that ketone group. Okay, that gives you 3-phosphoserine. 3-phosphoserine. Now you're going to sacrifice that phosphate there. The phosphoserine phosphatase takes the phosphate off, and now that's serine. So serine, essentially, it's 1, 2, 3 steps to get to serine from 3-phosphoglycerate. Then this reaction, which we've seen before, serine hydroxymethyltransferase uses tetrahydrofolate to take that methanol group there. You now get methylene, methylene tetrafolate, and water. And you make lysine that way. This happens in humans. That reaction, even though This one you don't need to know. But that's how you make serine and glycine from 3-phosphoglycerate. And these are the same processes shown up close and personally. Now, how does one make cysteine? You make cysteine from serine. Here's how you make cysteine. You have to activate this group here. So you can activate it with an acetyl group. So you serine acetyltransferase uses acetyl coenzyme A. That's a common metabolite. You activate this here like this. Then you take sulfide and you use O-acetylserine lyase. Sulfide attacks essentially here, and you attack here, and now you're going to liberate this group here. You liberate an acetic acid. The sulfide now converts this into serine. Cysteine. Now, how do you get sulfide? Here's how you get sulfide up there. This actually happens in our bodies. So you make sulfide from sulfate. Sulfate. So sulfate, in order to be activated, you use ATP. And you're going to take it, you're going to liberate the pyrophosphate. You're going to stick the sulfate right onto the alpha phosphate. This is called an adenosine biphosphorosulfate, or APS. This enzyme is called ATP sulfurylase. ATP sulfurylase. ATP sulfurylase gives you the adenosine 5-phosphosulfate. You're then going to activate this further by putting phosphate there. That's essentially APS. 3-phosphoadenosine 5-phosphosulfate. That's APS kinase. APS kinase. Phosphate here that liberates an ADP. And now you are going to subject this to reduction. So you have PAPS, 3'-phosphoadenosine 5'-phosphosulfate reductase, uses NADPH, and then sulfide reductase takes the sulfite, reduces it to sulfide. That's the most heavily reduced form of sulfur. That's what's used essentially to make cysteine. That's how you take sulfate, convert it to sulfide. Sulfide is toxic. Right after it's made, it gets used very quickly. Yeah, there are some bacteria that use sulfide in the soil as an excellent source. So the organism Vegetula lives near rice roots, and the roots of the rice plant have microorganisms essentially that make enormous amounts of sulfide. The Vegetula takes the sulfide, uses the electrons to essentially make ATP through sulfide oxidation. And then that sulfide oxidation produces sulfate. That's an example where we're going to use right away. In other organisms, you make sulfide, you use it right away. In humans, that's the part you have to know, you're going to make cysteine for the degradation of methionine. Once again, serine ends up being a part of this. So in bacteria, you make cysteine from serine. In humans, serine still plays a role. So homocysteine, where does homocysteine come from? That's the adenosine. That's the adenosine. Right. So you combine this with serine, you make cystathionine. That's cystathionine beta synthase of a PLP containing enzyme. Cystethionine is then degraded by cystethiolinase or cystethionine that gives you cysteine and alpha-ketobutyrate. Alpha-ketobutyrate is converted to propionyl coenzyme A. That's how you make cysteine. All right, let's talk about the aspartate family first. Aspartate is made from oxaloacetate. You should know how it's made. You'll need to know how asparagine is made. The others are members of the aspartate family, so that's K-mint. K-mint. That's kind of like the aspartate family, K-mint. So the first enzyme is aspartokinase. That's going to be the main regulatory route, main regulatory choke point for the aspartokinase. You make aspartyl beta phosphate. You then are going to reduce that, and you're going to make the aspartate beta semialdehyde. And now you have a branch point between the two. One of these is going to make lysine, the other is going to make methionine. Homoserine dehydrogenase will make homoserine. Dipicolinate synthase will make dipicolinate homoserine. That's the first step in the synthesis of methionine. That will be right to our choke point for methionine synthesis. Dipicolinate synthase. That's going to be the choke point for synthesis of lysine. Let's do lysine first. So you take your semialdehyde, and then you add pyruvate to it, and this gives you an unstable intermediate that's going to cyclize. So this is going to cyclize here, there. Make dihydropicolinate. Dihydropicolinate is used in trifosate bacteria to strengthen the wall of the endospore. This is how it's made. This pathway is different than the way you make lysine and fungi. Guess how you make lysine and fungi? You use alpha-ketoadipate. You essentially reverse the pathway by which you make it, by which you degrade in fungi. So bacteria use the DAP pathway, the diaminopicolinate pathway, and the fungi use the alpha-ketodipate pathway. All right, so dihydropicolinate, as you can see, is an oxidized. That gives you pyridine. So you get rid of that double bond. That gives you the pyridine to cycloxylate. You add succinate to it, that opens the ring. You add succinyl to aminoketoalpinolate. And you have an aminotransferase. You're going to put the amino group here, just like that. This gives you N-succinyl LL diaminopamellate. Diaminopamellate is used in cell walls of some bacteria. That's how you make it. Some bacteria use lysine as the number 3 amino acid. Some use mesodiaminopamellate. It's made in this pathway here. Others will use lysine. Others use mesodiaminopamellate. They're just one step away in the plasmacytic pathway. So then this undergoes desamination. The succinate is locked off with this hydrolase here. It's given this ridiculously long name. Succinyl diaminopamellate desuccinylase. That gives you the L alpha epsilon diaminopamellate. You then use an epimerase, and that epimerase is going to rearrange these two. So this gets put around here. So that's meso diaminopamellate E. coli. And other gram makers use this as number 3 amino acid in their amino acids of the peptide glycan. Make lysine, you just get rid of this carboxylate group here. The diaminopamellate decarboxylase, boom, there's your lysine. That's how you make lysine. That's pretty universal, gram-positive, gram-negative bacteria. What about methionine? You make homocysteine. So you take this, you're going to reduce it, make homocysteine. You then add succinate again on this side too. Succinate's used to activate these. That gives you succinylhomoserine. And look at this, cystathiamine gamma synthase. You're going to make cystathiamine. So cysteine comes in. Cysteine is made from the breakdown of methionine. Here you use cysteine to make methionine. You make cystathiamine, and then you have a cystathiamine beta lyase that gives you homocysteine. You add butyrofolate, you have methionine synthase, boom, you make methionine. What about tyrosine? Tyrosine is a branch from synthesis of methionine to use homoserine. So this will be, this enzyme here is used to control the synthesis of both threonine and methionine. So you put a phosphate on here, homoserine kinase. You then lop that off essentially, but the alcohol gets transferred here. That's threonine synthase. Boom, you make two steps essentially to make threonine. That's the aspartate family. Questions? All right, your handout has the next pathway I'm going to show you. I think it's on the first page. So alanine just gets made by transamination of pyruvate, using glutamate as the donator. That makes alanine. And then pyruvate will be used to make these two branched chain amino acids. These two branched chain amino acids, valine and isoleucine, are going to be made in parallel. The same enzymes will catalyze the corresponding reactions. So the first enzyme is going to take two pyruvates, link them together, and make alpha-acetylactate. That's alpha-acetylactate synthase. Alpha-acetylactate, however, can also be used to condense pyruvate with alpha-ketobutyrate. Alpha-ketobutyrate comes from the dehydration of threonine. You dehydrate threonine, giving out the amino group 2, you make alpha-ketobutyrate. Acetylactate synthase can also condense pyruvate and alpha-ketobutyrate. In one case, you make alpha-acetylactate. In the other case, you make alpha-acetyl hydroxybutyrate. And already you can see where this is starting to go. One's going to look a lot like valine, the other's going to look a lot like isoleucine. All right. These two are then going to be worked on by the same enzyme. Acetohydroxy acid isomeral reductase. So this will rearrange the alpha-acetylactate, and then it is going to be reduced. Here you get the dihydroxy isovalerate. Dihydroxy isovalerate. Now, if you give up a water and then stick an amino group on here, you have valine, don't you? If you do the same over here, you have isoleucine. So these two go through these parallel processes. Over here, you make dihydroxy methylvalerate. You make dihydroxy isovalerate. And this is going to take the acetohydroxybutyrate, convert it to the dihydroxy beta methylvalerate, same enzyme. Then these two are subjected to dehydration. Oh, dehydration. But I tell you, they got water. Now you make keto beta methyl valerate, and you make keto iso valerate. Now you've set up the ketone group for transamination. Valine aminotransferase will handle both keto isovalerate and keto methyl valerate. Here you make isoleucine, here you make valine. Examples of enzymes essentially that have active sites that can accommodate different substrates. So that's going to be the regulatory point for the synthesis of those amino acids. That's isoleucine and valine. What about leucine? Leucine's made from valine. And the genes involved in this synthesis, they form an operon called the ILV, isoleucine valine leucine operon. So valine is going to be converted to the first S intermediate alpha keto isovalerate. That gets shunted off essentially to make so-called isopropyl malate. So isopropyl malate synthase takes that to A. You stick an acetyl group on this. You stick the acetyl group on there and make isopropyl malate. Isopropyl malate is then going to be rearranged, and you make beta isopropyl malate. Then you're going to oxidize it. By oxidizing, you get rid of this carboxylic acid here. There, you make alpha keto isocaparate. When you have leucine aminotransferase, the enzyme has lots of homology to valine alpha aminotransferase. Boom, you get leucine. Once again, glutamate is more donator.
And that aminotransferase also can turn off the phosphate.
Yep. Yep. And now we go to the aromatic group. This group is nasty. This group, you talk about burning the midnight oil. I burned a lot of midnight oil to get this one done. Oh my word, this one took me forever. So again, just remember, it's not only bacteria that do this, it's also plants. Okay. So you start with erythrose 4-phosphate and phosphoryl pyruvate, and you're going to use this enzyme here that's going to make the 2-keto 3-deoxy arabinoheptulose 2-leucimate 7-phosphate. So this is a 7-carbon ditty, 1, 2, 3, 4, 5, 6, 7. And that is the KDAHP synthase. KDAHP synthase, that's usually what it's called. Which makes this monstrosity here. You're then going to get rid of this phosphate here. This enzyme has an NAD down to it that it uses, but it doesn't participate in the reaction. When you take the phosphate off, this is going to cyclize. So this will end up attacking up here. And you make the D3-dehydroquinate. And then that's going to be dehydrated. You're going to make 3-dehydroshikimate. And then you're going to reduce that and you make shikimate. Shikimate is an important control entity because shikimate is going to be used. Well, okay. Then you have shikimate kinase. Shikimate kinase is then phosphorylated. Well, shikimate phosphorylated makes shikimate 3-phosphate. Now you're going to add a phosphoryl pyruvate. There's where phosphoryl pyruvate comes in. You make 3-enol pyruvyl shikimate 3-phosphate. That is the 5-enol pyruvate 3-phosphate synthase. This is the enzyme that's inhibited, like glyphosate. Glyphosate you probably know as Roundup. Roundup. So that's how you kill weeds, is you inhibit that enzyme there. Then you're going to get rid of this phosphate and you make this compound here called chorusmate. Chorusmate. That's an important branch point then. Now, chorusmate is going to be used to make tryptophan. Tryptophan. And tryptophan, as you can see, is going to be therefore very dependent upon that aromatic pathway to send it chorusmate. So chorusmate is going to be aminated. You get rid of this as pyruvate. You make anthranilate. You're then going to build something that will make the endo group. So you add PRPP. This comes from the nucleotide biosynthetic group. And you make this 5-phosphoribosyl anthranilate. That enzyme is anthranilate desoxyribosyl. Then you have a isomerase, and that converts this into this forbidden enol carboxyphenol deoxyribose phosphate. Then you have the endo 3-glycerol phosphate synthase that makes the ring essentially. Now you have endo 3-phosphate, endo 3-glycerol phosphate, and then you use tryptophan synthase. This is a 2-part reaction that follows a ping pong mechanism. You have first, you give up the glycerol 3-phosphate, serine then binds, and you give up the water, and you make tryptophan. The tryptophan synthase is an enzyme that has a tunnel, has a substrate to essentially bind, and the intermediates go through the tunnel, interacting with it, and then block the other way. Very interesting enzyme. And synthase. And this just shows you essentially the mechanism of the tryptophan synthase. So the alpha subunit does this first reaction, the beta subunit does a second reaction. You get this amino acrylate adduct, the quinoid intermediate, then the albumin, and then boom, there you are. Very interesting enzyme, very heavily studied. Very interesting. Blah, blah, blah. The punchline is okay. Chorusmate's also the branch point for the synthesis of either tyrosine or phenolphthalein. So chorusmate is subjected to a mutase. Chorusmate mutates. That makes prephinate. And then these two steps here, then this will be inhibited by phenylalanine. This step will be inhibited by tyrosine. That's how you prevent your production of the two. Both of these will feedback inhibit on the chorusmate mutase. So if you make tyrosine, prephinate is subjected to a prephinate dehydrogenase. You oxidize it to 4-hydroxyphenylpyruvate. What you need to do is put an amino group there and a aminotransferase will do just that. The same amino, the aromatic aminotransferase will do this here. Yes, this is a PLP containing enzyme that makes tyrosine. Over here, you have prephinate dehydratase. Then using phenylpyruvate, you just put the amino group there in the film. And we talked about these already. These are steps of reactions we've already gone over. I remember tyrosine is used to make dopamine, norepinephrine, epinephrine. Histamine is made from histidine, one step. Take the carboxyl group out, there's cysteine. We looked at serotonin synthesis last time from tryptophan. Serotonin's then used to make melatonin, the 2 steps. And then glutamate is used to make amino, amino butyric acid with meth.
I wish we had more.
Now comes histidine synthesis. There's histidine synthesis. Remember, histidine's made from ribose 5-phosphate. Ribose 5-phosphate. So here's the first step. This is ATP phosphoribosyl transferase. ATP phosphoribosyl transferase. You take your phosphoribosyl 1-pyrophosphate. You're going to add that to ATP essentially. So your ATP is here and your nitrogen, as you can see here, is going to be bound to this carbon right there. These two essentially going to link up. So that's ATP phosphoribosyl transferase. Histidine is a non-competitive inhibitor of this enzyme in a manner that shows classic non-competitive kinetics. All right, then those 2 terminal phosphates get hydrolyzed by the phosphoribosyl ATP pyrophosphohydrolase, and you make phosphoribosyl AMP. And then you're going to cleave one component of the ring by adding a water across it, and that is phosphoribosyl AMP cyclohydrolase. And then you make this thing with a forbidding long name. Phosphoribosyl form amino 5-amino imidazole 4-carboxamide ribonucleotide. It's just EGADs. And then you subject that to an isomerase. Now, in E. coli, the cyclohydrolase and the isomerase, the pyrophosphorylase and the cyclohydrolase actually form one protein. It's a bifunctional enzyme that's 203 amino acids long. But in other bacteria, they have 2 separate enzymes, 2 separate polypeptides that do the same thing. All right, so the next enzyme is going to rearrange this. This is called an Amadori rearrangement. Amadori rearrangement. Yep. I think he was, yeah. And that gives you the phosphoribosyl form amino 5-amino 1-phosphoribosyl 4-imidazole carboxamide isomerase. That's the enzyme. And this enzyme is typically abbreviated ProFar. ProFar.
Yay.
Yeah. So that's going to do this. And now you're going to take an aminotransferase. You're going to add an amino group when you add the amino group, this molecule breaks up and you make this 5-amino-imidazole 4-carboxy ribonucleotide, also known as ACAR, and ACAR goes to purine biosynthesis. That's an intermediate in purine biosynthesis, which you will have to know. Anyway, by giving this group here the amino group, you're going to cyclize here. And now you make the imidazole 3-glycerol, imidazole glycerol 3-phosphate. Glutamine is your source of the amino group there. And in E. coli, the isomerase and the aminotransferase form one polypeptide, and they're often called IgP synthase together. And then you take out a water, and what do you see if you take out a water? So this is a imidazole glycerol 3-phosphate dehydratase. This is a manganese-containing enzyme. You can now put an amino group across that ketone. You have an aminotransferase. You have histidinyl, phosphate aminotransferase, make histidinyl 3-phosphate. Take off the phosphate, you have histidinyl. And then all one needs to do is to oxidize that, and you make histidine. Histidine. And in E. coli, the dehydratase and the phosphatase are encoded by the same gene. That's histidine synthesis, comes from ribose 5-phosphate. All right. This is a good place to stop. So I'll stop right there. We'll pick this up next time. We'll almost certainly finish this next time. And then we will move from there into the control of nitrogen metabolism. We're going to see that glutamine synthetase is the nexus of control of nitrogen metabolism. We'll look at nitrogen fixation, very interesting process biochemically, and then we'll move into nucleotide biochemistry and nucleotide synthesis after I record the scores.